I'm Mariana Wolfner. I'm the president of the Genetic Society of America. It's my honor to introduce our speaker today, Luciano Marraffini. He is giving the final lecture in the 2024 GSA Awards seminar series. So this past March, at the Allied Genetics Conference, Luciano received the Genetic Society of America Medal. This medal was established in 1981 to recognize society members who exemplify the ingenuity of the GSA through elegant and highly meaningful contributions to modern genetics. It honors individual members for their work in genetics at the mid-career level or up to 15 years since starting an independent position. Our speaker today, Luciano Marraffini, is the Caden Family Professor at Rockefeller University and an investigator at the Howard Hughes Medical Institution. Among his awards include membership in the National Academy of Sciences, the American Academy of Microbiology, and the American Academy of Sciences. Luciano determined how CRISPR-Cas systems destroy genetic targets with precision. This paved the way for genetic engineering technology development. His work unraveling the molecular mechanisms of CRISPR-Cas response is remarkable, and it's a feat that will long be recognized as a key piece of the paradigm-shifting introduction of CRISPR-based genome editing into biomedical research. Luciano explores genome editing and other potential applications for CRISPR-Cas systems, he ultimately hoping to answer fundamental questions about how these systems destroy their targets, how genetic memory is generated, and how the CRISPR-Cas immunity affects the evolution of bacteria and archaea. Luciano's lab uses Staphylococcus epidermidis and Streptococcus pyogenes as model systems to study CRISPR immunity. His current research uses molecular, genetic, and biochemical approaches to analyze the genetics and function of CRISPR-Cas systems, performing detailed analyses of the underlying molecular mechanisms of CRISPR immunity and facilitating the manipulation of this natural pathway for the development of new technologies. In addition to his research, Luciano's mentorship of PhD students and postdocs and the appreciation demonstrated by his mentees speaks to an invaluable investment in the next generation of scientists and the future of genetics research. Luciano's mentees describe his ability to make the lab a stimulating and fun place to do science and to interact with and inspire scientists of all career stages, encouraging them to take new risks and try new methodologies. So in a moment, I'm going to turn this over to Luciano to tell us about the synergy between CRISPR-Cas and restriction modification systems. But before I do so, two quick housekeeping items. The first is that all GSA online events are covered by our code of conduct. A link for that is available in the chat. The second is to ask you to put your questions for Luciano into the chat. We'll get through as many as possible in the Q&A portion at the end of his presentation. So with that, Luciano, welcome and congratulations again on the GSA medal for your beautiful work, which we are looking forward to hearing about now.
Well, first of all, thank you, Mariana, for the nice introduction, very complete, and thank you to the society for giving me this very, very prestigious award. I'm very honored to be one of the recipients. Considering the past recipients, it's really an incredible honor. So I'm gonna tell you today a little bit about the past of what I've done with CRISPR, how I kind of started. And then in the second part, I will tell you about this synergy between CRISPR-Cas and restriction. And just to give you just a starter, I like to do the New York Times crossword puzzle. And one day, this cue came up with this solo of Star Wars, this is in the popular domain, and I think most people know that the answer to this is Han. But then the next cue was what CRISPR technology can help to edit, and the answer for that is DNA. So what I'm getting at here is that this was a year ago or so. I realized that CRISPR has entered the popular domain. I guess people should know about CRISPR as they know about Star Wars, and that it was kind of very amazing to me since I started this so many years ago. But the reason for that is obviously because of the technological applications of CRISPR. In the last decade, there has been a gene editing revolution that has been fueled by CRISPR RNA-guided nucleases that enable this gene editing. This is not new. Probably a lot of people know that in the 70s, there's been another or a prior technological revolution or genetic revolution, and that was the recombinant DNA revolution. And this has been mediated by other type of nucleases, the restriction nucleases. And what restriction and CRISPR have both in common is that they're both phage defense mechanisms or defense mechanisms that bacteria use to prevent phage infection. And bacteria need really a lot of these anti-phage systems because phages are really bad, really predators of bacteria. And just for you to have an idea what I'm talking about, I'm going to show you a short video of cells being infected by phage. In this slide, I don't know if you can see my cursor, but I'll use my cursor. You have the 6 bacteria, which are Listeria monocytogenes, and the green dots represent phages that have been labeled with cyber green. This means that every cell that you see in this slide, in this picture, is being infected by one phage. And this is what happens with the cells. They grow a little bit and then they pop, they explode. And that just to show you how potent pathogens are viruses and why bacteria have developed many ways, not just CRISPR and restriction, to prevent these infections. So I've been working with CRISPR for more than 15 years now and giving seminars. And from the very beginning, there was always the same question at the end of all seminars, because people was very well aware of restriction nucleases because everybody used them for cloning. And there was all the time this recurring question of are restriction systems and CRISPR system related? They both protect bacteria. Are they synergistic? Do they interfere with each other? So it took us a long time, but we finally found an actual connection between these two that I'm going to tell you in a minute. But first, I'm going to introduce CRISPR to you. And CRISPR stands for Cluster Regular Interspace Short Palindromic Repeats, which is a long acronym, but it represents very well the architecture of these loci. These loci are in essence clusters of repeats, the first and the last letter of the acronym. And in this cartoon, which represents the CRISPR locus, the repeats are these white boxes. And the repeats are short, they're about 30 to 40 base pairs long, and they're actually semi-palindromic. But the most important feature of these repeats is that the repeats are not adjacent to one another. They're separated, they're regularly interspaced by sequences that are also short, 30 to 40 base pairs long, and that we call spacers. And their sequence is completely different. It has nothing to do with the repeat sequence. And I think in 2005, it was a kind of a key moment for the CRISPR field, because bioinformatic analysis performed by 3 different groups at the same time were able to blast these spacers and find that hits on the genomes of plasmids and phages that infect bacteria. So at that time, it was proposed that CRISPR will have some function in preventing the spread of plasmids and the propagation of phages. And indeed, later experimentation based on that hypothesis proved that the spacers in a CRISPR array provide sequence-specific immunity. If there's a phage that is infecting a host bacterium, that contains a CRISPR locus and a spacer that matches the genome of that phage, then that bacterium should be immune to that phage. And the way CRISPR works is basically in two stages. The first stage is, we call it immunization or generation of memory, is it involves the process by which the spacers are acquired or inserted into the CRISPR locus. And this was figured out by some of this first wave of experimental biology after the bioinformatic predictions by Barangu and Horvath. And they found that upon infection, CRISPR systems actually are able to capture sequences from phages and expand the CRISPR array. The new spacers always come as the first spacer of the CRISPR array. And this is a creation of a memory of infection, is a way of seeing this process. And then the second stage of CRISPR immunity, we call it targeting, and it's how this memory of infection in the form of a spacer is actually used to destroy a recurring phage or a recurrent infection. And the process by which this happens is, first the spacer is transcribed and processed into a short RNA that we call the CRISPR RNA, cRNA. And this turns out to be an RNA guide for some of the Cas ribonucleoprotein Cas nucleases that use the guide RNA. And I forgot to mention that the CRISPR array is always flanked by a set of CRISPR-associated genes that we call Cas for short. And among these CRISPR-associated genes are the Cas nucleases that use the CRISPR RNA as a guide. And they use the guide to find complementary sequences in the nucleic acids of invaders, such as plasmids or phages. And when they find this complementarity, then the nuclease will destroy and cleave the DNA. And this part, particularly finding that DNA is the target of many CRISPR systems, is something I did as a postdoc when I was in Eric Sonheimer's lab at Northwestern University a long time ago. And what we did to demonstrate that was, at that time in the mid-2000s, RNA interference, if you remember, and we still is very useful, is that it was a technology that was very in fashion, very used by the whole scientific community. And RNA interference uses short RNA guides to actually find complementary transcripts and destroy the transcripts. So the idea at the moment was that CRISPR will be kind of the RNAi of bacteria and use the guide RNA to find target RNAs. And so we will have these two possibilities, that the guide RNA is used to recognize the DNA or the RNA of a phage, for example. And what we did to test those two options was to insert a self-splicing intron. And in this way, the target on the DNA molecule is interrupted by the intron, and therefore the guide RNA doesn't have any more full complementarity to that the DNA of the virus or the plasmid. But after transcription of the DNA and self-splicing, the intron goes away and the target is reconstituted. So then in this setup, the RNA will have an intact target, and the DNA of the plasmid that we tested wouldn't have an intact target. And what we saw is that CRISPR immunity was eliminated when we had the intron. And this and other experiments that we did that allowed us to conclude that CRISPR immunity requires an intact DNA target rather than an RNA target. And that actually led us to finish the paper with some sort of prediction that, from a practical point of view, having the possibility to destroy DNA in a sequence-specific manner that is kind of programmable, like a programmable restriction enzyme, it would be very useful if the system could be transported into outside of the native bacterial or archaeal context. And that's what happened during the CRISPR revolution. But before that happened, a lot of work had to be performed to understand how really CRISPR works more than what we did by genetics. More biochemistry was required. And that basically is the characterization of Cas9 and how Cas9 cleaves DNA. And that was done, obviously, famously by Down and Charpentier, and then also by Sylvain Moineau in Quebec and Virgil Signis in Lithuania. Altogether, the work showed that Cas9 uses a guide RNA to find complementary DNA and then cleave that DNA, cleave both strands of that DNA, generating a blunt end or a double-stranded DNA break on the DNA. And then kind of with that information, I collaborated with Fang Zhang to determine whether CRISPR can actually be transported into human cells and cleave human DNA, leading to gene editing. And we did that. We transported the Streptococcus pyogenes CRISPR-Cas9 system. We were one of the first two groups that did it. We and George Church, also at Harvard, published together with us at the same time. We transported the Streptococcus pyogenes CRISPR-Cas9 system into human cells. And when we expressed Cas9 and the guide RNA, we found that that was sufficient to create some indels at the site of cleavage of Cas9. I'm not going to go through details because it's in the interest of time, but we saw a modest but noticeable gene editing phenotype that again is CRISPR will cleave, and then the DNA will be repaired, and when the DNA is repaired in human cells, indels, insertions or deletions of a few base pairs are generated, and usually that is used to knock out genes. We also did some homologous recombination experiments when we put a template for homologous recombination to repair the cleaved DNA, and also we saw, in this case, we put a HindI3 site and NAH site, which are restriction enzymes. So everything kind of will become circular. But and then we saw that we were able to introduce those sites in human DNA. And since then, I think that the technique has been picked up by a lot of labs around the world, and a lot of it's not used against just in humans to do gene editing, but also in many different organisms. So let me now introduce to you the restriction systems very quickly. Restrictions are also enzymes that cleave DNA and generate, for the most part, double-stranded DNA breaks, but they do so without a guide RNA. They cannot be programmed. They have an active site which will recognize a specific sequence, and that sequence cannot be changed. But phenotypically, so to speak, the outcome of restriction is very similar to the Cas9 outcome. It's cleavage of the incoming phage DNA. Now, a particular issue of restriction is that the restriction site that is recognized, which is 6 to 8 base pairs most commonly, or 4 base pairs. So those restriction sites, the sequences are present many times, usually in the host chromosome. So restriction enzymes or restriction systems have to avoid the autoimmunity, so to speak, by working together with methylases. So the methylases will methylate the same site that the restriction cleaves, the restriction enzyme cleaves. And by methylating those sites, they make those sites refractory to cleavage by a restriction enzyme. And that's how the chromosomal DNA is paired from nuclease activity of the restriction enzyme. And usually the phage DNA will be, at least at the beginning of an infection, will come from another organism, or but will come not methylated, so it will be restricted. But of course, you can imagine that methylases by chance sometimes will get to the restriction site before the restriction nuclease gets, and it will methylate. They will methylate the restriction site on the phage DNA. When that happens, the phage DNA becomes now refractory to restriction, and therefore the phages can escape. And this happens quite frequently. It's very common that, and also because you only need one phage that is methylated, that it will start spreading through the population, and in the end it will bypass the defense that restriction enzymes provide. So what we found, and given what I told you, these are the two phases of CRISPR immunity, what we found is that actually restriction really helps with the immunization stage of CRISPR, which is something that was a little bit less intuitive than the other possibility that is synergistic with the targeting. And to explain the results, I first need to explain how we measure spacer acquisition in the lab and in the field in general. And it's a simple PCR experiment where you put a forward primer that is upstream of the first repeat, and a second primer, a reverse primer on the first repeat, on the first spacer of the array. And when you get bacterial DNA and do this PCR, you get a small PCR product. But then after infection, many of the cells will acquire a new spacer. And because it goes into the first position of the array, when you perform the same PCR again, then you get expanded or larger PCR products that you can see here. And these PCR products, the beauty of this is that they can be cut from the gel and extracted and then sent for deep sequencing. And then you will have all the sequences that were acquired from the phage genome and also kind of the number of reads for that sequence. And I have to mention that this CRISPR immunization or spacer acquisition is not a very common event. In most bacterial system and in ours, for sure, we measure it as about 1 cell in a million cells acquire a new spacer. And so that's why we need this PCR amplification to actually detect it. So with the deep sequencing information, we can determine where the spacers come from the phage genome by just looking at the sequence of the spacer. It can be in the middle, it can be in one end. The spacer can be acquired from the other end, but also by the number of reads for each spacer sequence, we can get an idea of how often these spacers are acquired. Some spacers are acquired very frequently, and some less frequently. And then we can do plots of that describe the spacer acquisition pattern across the phage genome by plotting each spacer on its corresponding phage target sequence with the number of reads that we get for each spacer, and then we get this again the pattern of spacer acquisition. So what I'm gonna tell you now is work that's been done by a student in my lab, a former student, Pascal Maguin, that actually when he started on this question of what is the relationship, if any, between CRISPR and restriction. And we work with staphylococci, as Mariana mentioned, and we couldn't work with the endogenous staphylococcal restriction systems because they had a lot of restriction sites in the phages that we used. So we decided to put into staphylococci a Bagel2 restriction system. Bagel comes from Bacillus, and it has a simple restriction site of 6 base pairs. One of the phages that we used in the lab had only one site, a Bagel2 site, and that was very handy. And what Pascal did was to either erase this site, made a mutation on the site, such as to work with a phage that doesn't have any restriction sites, but also add additional restriction sites by engineering the phage. And then he did the spacer acquisition experiment, and just by looking at the number of sequences and reads that he got, there was an immediate correlation between the number of restriction sites on the phage and the number of spacers, or the percentage of spacers that were acquired. You can see that with the phage that where the restriction site was erased, and so there has no restriction site, there's very little spacer acquisition. With the 3 phages that have only one restriction site, it's about 5% of spacer acquisition. And then with the phages that have 2 restriction sites, then this increased by 15 or so percent. So there's a quantitative effect of having restriction and CRISPR, that restriction helps CRISPR, enhances CRISPR acquisition. And then when he did the pattern, when he plotted the data, the next generation sequencing data, here, for example, is the comparison between, in black, the phage that doesn't have any restriction site, so it was not restricted by bagel, and in blue is the phage that have one restriction site at the A site. And you can see that without restriction sites, there's a very noisy pattern, but in the presence of a restriction site, there's a very big peak or a prominent peak that is at the same sequence that is, of the restriction site. And he corroborated this with the other engineer phages. And you can see here now the phage that has the A site is erased and the C site is inserted at a different place. You see a peak at the C site, and the same is true with the third phage that has a restriction site on the B site. And then with the phages that have two restriction sites, it's the same. The peaks are maybe less prominent because now they divided into two restriction sites, but you can see the yellow is the AC, and you see one peak here and one peak here in the C site, and then the orange is the AB phage with two restriction sites, and the two peaks are again in the site of restriction site A and restriction site B. So it turns out that restriction helps quantitatively, but qualitatively, the reason why restriction helps is because, and I'm not going to show you this data because it's data that has been we figured out this through many years and many studies. The CRISPR acquisition machinery, the integrases that grab phage DNA and insert it into the CRISPR array, they really prefer free DNA ends to do so. And the restriction nucleases actually create two free DNA ends. And the restriction enzyme, the acquisition machinery actually uses both ends as substrates for spacer acquisition preferentially. So that's why we see a peak at the restriction site. And then that's in terms of the mechanism at the molecular level. That's why restriction enhances spacer acquisition for CRISPR. But at kind of at the population level, what happens is we studied that as well, and you can see here this is a curve over time at zero of the growth curve of the culture that has been infected with phage at time zero. And both cultures have a CRISPR, which doesn't have any spacer against the phage. It's a naive CRISPR system. And then we have the blue culture, it has bagel2, and the red culture doesn't have bagel2. And immediately you can see that in the end of the experiment, after 1 day of incubation, the cells that actually are able to grow back or to grow to survive phage infection are the ones that contain bagel2. And we did this PCR analysis of the CRISPR array over time, over the course of this experiment, and you see that at the end of the experiment, all of the cells that survive actually are, so these are the lower PCR product is the CRISPR array without a new spacer. And by the end of the experiment, you see that all cells in the population contain new spacers, because that's what allowed them to survive. In contrast, when there was no bagel2, you see a little bit of spacer acquisition because the cells have CRISPR, but that this was not sufficient, at least in this time frame. And at this MOI, multiplicity of infection, it was not enough to allow the culture to survive phage infection. So what we think is happening here is the following. The restriction provides some initial protection, and we can see that from the first couple of hours of the growth curve, you see that the cells with bagel2 keep growing for the first 2 hours, but the cells without the restriction system, they collapse quicker. And that's the immunity that restriction is providing. And you can actually, during this time, although you don't see bands for spacer acquisition in the gels, because I told you that it's very few cells acquire spacers. When you do deep sequencing, we did deep sequencing at different times during infection, you can see that the culture with bagel2 had always 1 log more of spacers acquired, that an order of magnitude more than the acquired spacers in the absence of bagel. So in these first 2 hours of infection, the presence of bagel 2 allows the acquisition of more spacers. And this will be important later, we think. Eventually, the cultures, even in the presence of bagel 2, they collapse. So they go back to very low OD and this is because the phage got methylated and started spreading and bypassing restriction. And we can measure that. We did an assay to calculate what the fraction of the phages at a given time point are actually modified. And we found that by 1 hour, you can see here in this graph, by 1 hour in the cells, in the population, the culture that had bagel 2, all the phages that we were recovering from that culture were modified already. So that's the reason why we think that this culture collapses, because the phage got methylated. But then, obviously, at the end of the curve, the cells can pick up, and that's when we think that the initial immunization that happened thanks to restriction that enhances CRISPR spacer acquisition, those cells are able to fight phage infection, but they are very few. So it takes a while, until several hours, until those cells actually become the majority of the population, and then they allow the OD to grow, increase. And we can see that again in the PCR products, because the first spacers that we don't see here, but we saw by deep sequencing, that are acquired in these early hours of the experiment, they are basically, they're all the cells that acquire spacer now are selected, and the only ones that survive at the end of the population, they have acquired spacers. So this is more or less what the model that we have is, that restriction works as an initial response, and actually the cleavage that it generates, the cleavage of the phage DNA, it generates the substrates for spacer acquisition, and that allows the CRISPR system to acquire spacers. At that point, at that initial stage, the spacers are not really required because the cells are protected by restriction. But then when restriction fails, those spacers actually come very handy, and there's a kind of a second or late response that it uses the spacers and CRISPR-Cas9. And you can, I don't want to make a big analogy, but it's somewhat analogous to the initial response by restriction endonuclease is somewhat of an innate immune response. And the late response in CRISPR, because it actually acquires the memory of infection, it can be considered an adaptive response. And mammalian immune systems actually work a little bit like that. After first exposure, there's an innate immune response, but not so much of an adaptive immune response. And then after a second exposure of a pathogen, then the innate immune response is always the same, but the adaptive immune response is much bigger, and it helps prevent completely the disease by that pathogen. And the other concept that I think it's interesting is that not just restriction enzymes, but any nuclease, because of this property or feature of CRISPR systems, that they use free DNA ends as substrates for spacer acquisition. Every nuclease that will generate free DNA ends, it's a good, it will enhance spacer acquisition. And I don't have time to show you, but it's been shown by us and others that REC-BCD enhances, which is a nuclease involved in DNA repair, also generates free DNA ends when it degrades DNA, and it enhances spacer acquisition. And Cas9 itself, this is a very interesting phenomenon that we observe. Cas9 creates double-stranded DNA ends the same way as a restriction enzyme does. And those ends are used by the Cas9 system to acquire more spacers. So the system primes itself for the acquisition of more spacers. And the reason for this is because the model right now for how CRISPR works is that the first infection needs to be a defective infection in order for the cell to have time and don't die from infection, and then acquire the spacer and fix it in the population. If the cells that acquire spacers are killed by the phage that is infecting them, then the spacers will never be present in the population. So all of these nucleases actually they have a two punch on the phage. They make it defective, so the phage will inject its DNA into the host cell, but the DNA will be inactivated, so to speak. The lytic cycle will not continue, but the DNA is still there. And it's also cleaved, and it has two free DNA ends or free DNA ends from which the CRISPR system can acquire. And so that's kind of the two functions of nucleases in enhancing spacer acquisition. So with that, I'd like to acknowledge, obviously, the people. This is the lab right now, and Pascal has left. And Pascal did all the work on restriction and CRISPR synergy. And then the funding, Rockefeller, Howard Hughes, NIH, and the Kaiden family, and also past funding, and also GSA, again, for giving me this tremendous honor. I really appreciate the society for selecting me for this award. And with that, I'll take questions.
Thank you so much, Luciano. That was really, really cool and really beautifully delivered as well. I'd like to invite anyone who has a question to put it into the chat, and then I will call on you in order. And again, if you would like the question asked anonymously, either send a private message to Jessica or to me, and we will ask it for you anonymously. So yeah, so any questions for Luciano? Well, while we're waiting for people to type, I have one question, which is, yes. If a phage has multiple places that could become spacers, it sounds like if one is near a restriction site, that would make it more likely to be acquired.
Yes.
Is there? Yeah,
that's correct. But one thing that, so I didn't show you the data, but and the reason why we put Bagel 2, which has only a single restriction site, is to be able to, because it's only one restriction site, we saw the peak. And that was very kind of useful to understand what was going on. When we use the endogenous Staphylococcus restriction systems, for example, this phage has 26 restriction sites. And so instead of just one site being most preferred, you see an enhancement of spacer acquisition, but it's all across. You cannot distinguish peaks when you have 26 restriction sites. And that's more normal, or in nature, that phages will have more than just one restriction or two. And so I think in the end it gets a little bit diluted. And the other thing that is very interesting is that every double-stranded DNA break in bacterial cells is processed by REC-BCD, which is a nuclease that choose free DNA ends. And REC-BCD, and I mentioned at the late, as a nuclease that enhances spacer acquisition. And the reason it enhances is that REC-BCD, once there's the restriction site here, REC-BCD will chew it and move the free DNA end far from that restriction site. And it will leave a free DNA end far from the restriction site. And so you will lose kind of the mark or the specificity of the restriction site for spacer acquisition, and then you start acquiring, the CRISPR system starts acquiring from different places of the genome.
So just a follow-up to that question. So I know that different guide RNAs are better or worse. Mm-hmm. Is there selection on having bacteria be selected for to have the better spacers with time?
So, okay, so there's two questions, two answers, or two things that I know that answered that. One is, for example, that spacers that are, so phages, double-stranded DNA phages usually enter as a single-stranded DNA.
Mm-hmm.
And there's an order of injection. An injection can last, for lambda, which is the only phage that this has been measured, injection can last between 5 and 15 minutes. It's been shown by some microscopy measurements. So what we found is that if the spacers target the first DNA that is injected into the cells, they can be, when you use very high MOIs, that matters. Meaning MOI is multiplicity of infection. When you have a lot of phage around, having a spacer that targets the first DNA that enters the cell is better than having a spacer that targets a late region of the phage. However, that is in the lab, and this is we after that study we tried to do bioinformatics to see whether that was a selective force, and we didn't see that in the natural spacers, and that it's somewhat difficult. Spacers, there are other selection events in natural populations that are just not just phage that may change the composition of spacers kind of randomly, and so RegBCD has no nucleus activity, also adds to acquisition. How do you suppose that works? So RegBCD actually has, in my understanding, RegBCD clips DNA, has two domains. One clips one end, one strand, another domain clips the other one. And it clips in a way that one strand is clipped more than the other to leave a 3' overhang for recombination. But we don't know exactly how that works. But what we see is that, so RegBCD also degrades DNA in this way up until it reaches a chi site, which is a sequence in the genome of bacteria or in the genome of the phage that makes RegBCD stop degradation. And so we see a high, when we do experiments to measure that, we did see the hotspot of spacer acquisition was between the restriction site, for example, and the chi site. So RegBCD enhances, that's what I was saying, it moves the site of acquisition from the restriction site to the first chi site, which is where RegBCD stops. And and the question is, how do you think that works? And it's it's only how I think it works. That has not been proven or demonstrated. But the idea is that RegBCD, although it's very processive, it might fall off in some cells and in some cases. And when it falls off, it will leave a free DNA end, and then the spacer acquisition machinery actually can acquire. But that's how we think it works.
Thanks for this beautiful talk and work. I wanted a couple of quick questions, I think. What governs the loss of memory to prevent phage bacterial genomes from increasingly larger? Is there a random loss with age, or is there a selective deletion of memory?
I think nobody really knows, but in the lab, we don't see spacer loss. Mm-hmm. But when you look at metagenomic data of populations in natural habitats, you do see that the CRISPR array is very variable. But what you see is that usually there's a conservation of what would be the 5' end of the CRISPR arrays. So in a population of bacteria, the 5' end of the CRISPR array contains the spacers that were most recently acquired. And those align and those are conserved. And then whatever is more on the trail end of the CRISPR is much more variable. And there are signs of deletions. And so it seems like the most recent spacers are conserved supposedly because the thread is still there doing the selective, pushing a selective force to maintaining those spacers. And then the spacers that are not used get deleted from the CRISPR array. And the thinking is that, which has not been proven yet, is repetitive loci, usually they can go through mistakes of replication because the replication machinery is copying a repeat, and then it jumps, I think it's DNA polymerase slippage, it jumps into another repeat. and then whatever was in the middle is lost. And if what that was in the middle is lost, those spacers are actually not providing defense, useful defense. There's no selection to keep them, and then they get lost. And I think that that's that's what we know about there's definitely shrinking of the CRISPR array. Although there's an archeon that has 580 something spacers. That's a record holder, which is quite a lot.
Yeah, good. Thank you. And I wanted to ask also if, I mean, this provides a new way of thinking about genetic recombination. Have evolutionary biologists gotten a hold of this and looked at the relative selective value for genes?
Yeah, so one of the earliest studies of CRISPR that really show kind of provided very strong evidence that CRISPR is providing defense against phages is that there's the recombination. They looked at, this is Jill Banfield at UC Berkeley, where she looked at archaeal populations that are very that are monocultures practically. So they're populations, but they're very few species, and the phages are coexist. And she could determine that the rates of recombination really are every 35 base pairs. And the thinking is, maybe I'm not expressing that right, but the thinking is that because the length of a spacer is 35 base pairs long, phages were recombining themselves every 35 base pairs to generate new variants. And if you send me an email, I can send you the paper. It's a very interesting paper that in a very non-molecular way, a very evolutionary way, proved that really CRISPR is defending against phage infection.
Yeah, thank you.
The next question is by Joana Carneiro da Silva. Her question is, seems like phages would have evolved larger genome sizes with junk pieces that have rapid evolution to prevent recognition, but that would come at a cost for replication time. Do you see evidence for this type of conflict in phage genomes?
Well, I think that I don't see evidence of that conflict, and I'm not also an evolutionary biologist, so I don't know for sure. But in a way, CRISPR will prevent that, will make that enlargement useless, because the adaptive fashion of CRISPR, if a phage will incorporate junk DNA to avoid targeting, CRISPR will get a spacer from that junk DNA. I think that's the nature of CRISPR, that it can get, it's not really, it works against any phage that injects DNA, and it can grab spacer from any region. So I think it wouldn't, in the face of CRISPR, that enlargement of the genome size will not really provide an advantage to the phage.
So Scott Emmer had a question.
Beautiful science, a wonderful seminar. A very simple question.
Thank you.
In theory, one could load spacer DNA from the host chromosome. Now, clearly that would mean destruction of the host chromosome, but is there any evidence?
Yeah, that happens all the time. CRISPR systems don't have a way to distinguish free DNA ends from the genome or the phage, and in fact, there's a tendency in chromosomes to have broken DNA at the end of at the terminus of replication, and all phage when you do spacer acquisition with very deep sequencing, when you can detect even low frequency events, and you do it in the absence of phage infection, just CRISPR native to an organism, you see spacers acquire, and you see them from the terminus of the chromosome of bacteria. Presumably, that's kind of an autoimmunity cost that CRISPR has. But those individuals of the population die from autoimmunity or self-acquisition, and they are eliminated from populations. You don't see spacers. It would be very, very uncommon to see spacers in natural populations that target chromosomal sequences. But in the lab, we detect them all the time.
Interesting. Thanks a lot.
So we have time for one more brief question, which is from Shuting Yu. Restriction systems help CRISPR. Do you know which was the evolutionary occurrence and fixation? Did restriction enzymes systems come before CRISPR and are more ancestral, or was CRISPR first? Is there any evidence for this?
I don't know that has been discussed. That's a question for Eugene Kooning, who's done a lot of great work on the evolution and of CRISPR systems. No, I know that there's many different CRISPR systems, and some of them seem to be more ancient than others, but I don't know. One thing is that restriction is much more widespread than CRISPR. CRISPR is about in 50% of the sequence genomes, even lower if you measure by, if you do kind of metagenomic sequencing of environments. But restriction is much more prevalent. And so maybe, I don't know, that doesn't mean that one is before the other. It's just one is more successful than the other, but I don't know the answer to that.
Thank you. I would like to thank Luciano again for really a beautiful seminar, a beautiful body of work, and to congratulate you again on the GSA medal, and to thank everyone for coming and for the excellent questions and discussion.
Thank you again, and thank you all for joining for the seminar.